Hello and welcome to Nikolai's Genetics Lessons, and in this video we are going to talk about the difference between paracentric chromosomal inversion versus pericentric chromosome inversion. So take a look at this picture, and this is picture of the human karyotype. You can see here 22 pairs of autosomes and also pair of sex chromosomes. If it's going to be X and Y, this is going to be male karyotype. If it's going to be 2 X chromosome, this is going to be female karyotype. Also in this picture you see dotted line, and this line shows where centromere in each chromosome we can find. And you can see, for example, in chromosome number 1 that centromere is exactly in the middle, but in other chromosomes it is closer to one tip of the chromosome, so dividing chromosome to two unequal parts with long arm and short arm. We specify them. P stand for the short arm and Q stand for the long arm. As you see, in other chromosome centromere is very close to one tip, and in some animals we also can find such chromosomes which consist of only one arm. Chromosomes would be connected at the very tip of chromosome, just giving a rise to chromosomes with one long arm. But human karyotype doesn't have such chromosomes, which we call telocentric. But in human karyotype we can divide all the chromosomes to short and long arm. We have trillions of cells, and each cell contains the set of the chromosomes. And of course sometimes mutations may happen. Usually they would be repaired, but sometimes they would be unnoticed. And some of them would be just point mutation, when, for example, just one nucleotide would be inserted, deleted, or just short fragment, but sometimes it can be a large fragment. And when we're using dye technique, like on this picture, we would be able to see such changes on the chromosomal level, because the pattern, expected pattern of the chromosome would be different, and we would be able to see such large mutations on the chromosomal level, for example, such as deletions, duplications, or inversions. Now I want you to take a look at this picture. Here we see two variants of the inversions that may happen. One variant when breakage would be in one arm. It can be long arm or short arm of the chromosome, but if it happen in the same arm, and then we have inversion here, as you see, yellow and green change places. So this fragment was inserted back but was flipped over 180 degrees. So we didn't lose any genetic material, but now it is in the different order. And in such large fragment of the chromosomes, we expect that there can be tens or hundreds of the genes. If such inversion happen in the same arm of the chromosome, we call this paracentric inversion. But if chromosome broke in one arm and also in another arm, and in the middle we have a centromere, then we call such inversion pericentric inversion. And this is all for today. Subscribe and see you in the next video. Goodbye.